Hi, my name is Morgan Yonke, and I did my paper over DNA polymerase. First, I wanted to talk a little about just proteins in general. Proteins are an essential part of all living organisms. They drive practically all biochemical processes. They allow movement of signals between cells and help us keep cells in their proper shape. They also have a unique amino acid sequence with 20 different amino acids that help make 4 different levels of protein structure, which are primary, secondary, tertiary, and quaternary. Primary structure is determined by DNA and uses peptide bonds. There's many different varieties of primary structure. Then secondary structure is the regular repeated folding of polypeptide chains. It can either be in an alpha helix or formed by hydrogen bonds or a beta pleated sheath. Tertiary structure is the full 3D conformation formed by an entire polypeptide chain using the alpha helix and beta pleated sheaths and any other loops and folds. The tertiary does include R groups and includes many bonds and interactions. Quaternary structure is just the same as tertiary, but with one or more polypeptide chains that come together to form a big protein. Next, I'll be talking about my protein, which is DNA polymerase. So DNA polymerase are enzymes, and they are in all living cells. You need DNA polymerase to survive. What it does is basically creates DNA molecules by building nucleotides, which are the building blocks of DNA. They work in pairs to create 2 identical DNA strands from one molecule. There's many types of DNA polymerase, some that they have known for a while, and some that they're just figuring out. Not a lot of mistakes are made with DNA polymerase because it does proofread the DNA after it's been copied, so that mistakes can be corrected. But if a mistake does happen, it could cause some serious consequences, which I'll talk about later on in the slide. So, polymerase structure. They like to call it the right hand of the cell because it resembles a lot of a right hand. In this diagram I put below, it shows the thumb, the palm, and the fingers. The thumb and fingers are in the same family, and the palm is in a separate family. The thumb and fingers do a lot of the positioning, and then the palm is used for binding. So the first DNA polymerase was isolated by E. coli, and that was a crucial step in determining all of the next new DNA polymerases. It was done 50 years ago by the work of Arthur Kornborg's laboratory, and very few DNA polymerase families were known back then, and now there's over 15. So there's a lot of diseases associated with DNA polymerase. Like I said before, they don't usually make mistakes, but if they do, you can imagine that it's going to make a big impact. So a lot of times DNA polymerase is associated with a lot of different kinds of cancers, but those are definitely not the only kind of diseases that DNA polymerase can cause. Things like Alzheimer's, it has been known to cause immune diseases and more things like that, but a lot of it has to do with different types of cancers. Also, it can be resistant to chemo, which has been a big problem. So this is a picture that I found in one of my articles. It's called Climbing Mount DNA Polymerase. I thought it was interesting because it just kind of basically goes through all of the years starting from 1956, from the discovery of the first DNA polymerase in E. coli, to present, that they're saying there's no new DNA polymerase found, but now we're just trying to basically figure out how the new families and the new types of DNA polymerase work and how they function. So that's about it for my PowerPoint, and thank you guys for listening.